Our lead molecule, IPI-145, is a very selective and very potent inhibitor of PI3K delta and gamma isoforms. It has shown remarkable activity in heme malignancies where we are testing the molecule in phase 1, and today we've announced the opening of a phase 2 trial in indolent non-Hodgkin's lymphoma, approximately 120 patients in the relapsed refractory setting. Our phase 1 experience had two portions: a dose escalation where we enrolled 33 patients between 8 and 100 milligrams. We defined 75 milligrams as the maximum tolerated dose. We opened cohorts at 25 milligrams in the second phase and 75 milligrams. Both doses are very potent, show very potent activity in blocking phosphorylation of AKT and downstream chemokine and cytokines. And both doses have shown a great deal of activity across a broad spectrum of hematologic malignancies, both in B cell and T cell. The data is still maturing, but we see responses of about 68% response rate in the early assessment of indolent non-Hodgkin's lymphoma, mostly follicular lymphoma, and we have seen response rates of 55% with an additional 33% nodal responses in chronic lymphocytic leukemia. It's a little earlier in the T cell malignancies, but we've seen very nice responses in patients with Sézary syndrome, as well as patients with peripheral T cell lymphoma. IPI-145 has been generally very well tolerated by patients. We don't see any dose-related adverse events. We have had sporadic adverse events due to comorbidities associated with the diseases like CLL and lymphoma, but this is to be expected in a very heavily pretreated phase 1 population.